Again, I want to thank everybody for joining us. Today's session is entitled Avoiding Outcomes Bias and Driving Quality Decisions in Biologics Development and is presented by IDBS. Today's session is also sponsored by Astrix Technology Group. Next slide, please. Before we get into the materials for today, I want to take a minute to, if you haven't been on any of the Astrix Partner Webcast Series webinars or any of our regular webinars, we'd like to just introduce you to our company and explain what it is that we do. First and foremost, we're an informatics professional services and strategic outsourced solutions company. We are dedicated 100% to servicing the scientific community. A couple of quick stats about the company. We were established in 1995 and are privately held. The company originated as an IT division of a firm called APBI Life Science, which was a research organization. 7 offices. Today we operate in the United States and beautiful Costa Rica, and our headquarters are in Red Bank, New Jersey. The companies that we support are Fortune 1000 life science enterprises, chemical, CPG, government and research institutions, mostly with large and fast-growing IT, outsource, and compliance needs. The mission of the company has and always will be to deliver scalable, sustainable solutions to the scientific community. Next slide, please. The group that has been hosting this partner webcast series, the webcast that you're hearing today, and the other ones that we've done, is being sponsored and put on by our professional services group. The professional services group inside of Astrix focuses on end-to-end services that span the complete life cycle of scientific data systems. So this means that the company engages in things like business process analysis and enterprise architecture, both assessments and development. What that means, a fancy term for, we come in and we figure out what technology you need, what your technology stack should look like, how that technology should be deployed based on the unique makeup and needs of your laboratory. And then we go through typically laboratory technology selection services. So once we've designed what we feel like your stack should look like, we will help you choose the right solutions in a very vendor agnostic way for you to make sure you're getting the right tools that you need for your unique environment. From there, we can certainly act as an agent to develop and implement that software. And then following that, we can certainly do a complete computer systems validation of that software. After the fact, we have 360-degree laboratory system services. What this means is we can help manage your laboratory systems either on your premise or in the cloud. And we can also provide scientific and technical staffing services. So if you need staff to take care of those systems, we have a large division of staffing and technical services folks that we can put in place to help you with that. At the end of the day, we evaluate, implement, and manage systems to enhance the collection and processing of scientific data. So that's just a little bit about Astrix. I'd like to now introduce you to today's speaker. So we can go to next slide, please. And Matthew Clifford is going to be presenting today. Matthew is joining us from the UK, and he's a senior product manager at IDBS. He brings 19 years experience working within and alongside the pharma industry, including 8 years in informatics roles in a top 20 pharma. Supporting discovery and preclinical organizations. He brings a proven background in industry trends and solutions. Here at IDBS, Matt leads the development of solutions to support all aspects of R&D across pharma, from bench, both within IDBS and in partnership with other life sciences divisions, Danaher Life Sciences divisions. He has a degree in chemistry from the University of Newcastle upon Tyne in the UK. So Matt, with that, I'm going to go ahead and turn it over to your capable hands and have you take us through the rest of the presentation. I'll come back at the end to help field questions.
Thank you for that introduction. And before I start, let me introduce a little bit about IDBS. So IDBS is a software and service provider that helps research and development teams and scientists in biologics development and drug discovery and development across different facilities around the world make discoveries that have the potential to transform the lives of populations worldwide. Our diverse customer list includes 22 of the top 25 pharmaceutical companies, as well as R&D driven organizations across a multitude of sectors. And our purpose is to provide the best-in-class cloud-based R&D technology solutions to the world's most forward-thinking companies, helping them to solve global challenges. For the last 10 years, IDBS has been working with the world's leading pharma and CDMO companies to help them to digitally transform their businesses. Over this period, we have developed a deep understanding of the complex processes and challenges that exist across the biologics lifecycle. It is through this that we learned that there were opportunities for us to better develop our customers, more quickly lay the foundations of a digital transformation, which will affect the time and the cost of delivering life-changing vaccines and therapies to patients. And it is with all this in mind that we set out to build a capability that helps eliminate manual, repetitive tasks with high throughput processes in biologics development, whilst providing rich but easy to use tools to accelerate data interpretation and decision making. This new offering is to be part of our approach to how we move customers from digitally enabled environments to a digitally transformed one. The biopharma industry has huge ambitions to accelerate the move towards Industry 4.0 approaches. That is smart factories, process automation, real-time data to ensure high quality and efficiencies through quality by design. Yet, frankly, much of the process today is disconnected with difficult to monitor offline processes, where the analysis of data and decisions is achieved using separated and discrete data sets. So in today's discussion, I will argue that this is leading to inefficiencies, rework, and poor decisions being progressed due to a lack of timely, available data to drive insight, which is in fact compounded by the out-of-date software tooling and manual transcription of data that regularly occurs across laboratories. But let's pause for a moment and just consider something a little different: outcomes bias. Outcomes bias is our tendency to judge a decision based on the outcome or result. Let's explore this together with two decisions and think about the impact of these two decisions. The first is decision A, and I'm going to read this out verbatim. You decide to invest in a biotech, as you know and trust someone who is working there. They tell you how great their company is, and the stock you invest into is a huge success. And decision B, you decide to invest in a biotech, as you know and trust someone who works there, and they tell you how great their company is, and the stock you invest into is a huge failure. Which of these decisions was better? A or B? The thing is, in neither case, A nor case B, did I give you any meaningful data to conclude. I only told you the outcome and a near identical description of the decision. Yet many people will tell you that decision A was a better decision because of the outcome. If you were basing an investment decision or a portfolio, you would want more data. What is the free cash flow of these businesses? What is the market for their research? What are the macroeconomic conditions that they're working within? And so on and so forth. In short, you would use more data to make this decision, to avoid trusting to luck. Because when you repeat the exercise, you may not get the same outcome. The hint here is to avoid using the quality of the outcome to validate the quality of the decision and to look to try and bring more data to bear. Perhaps a really silly example, but one that's very transparent and easy to grasp. You run a red light and you get to work 5 minutes early, and the outcome therefore is great. So you decide to make this your commuting strategy every day to work. Now, I think we can all instinctively tell that this is an unsafe decision. However, this is a mental shortcut that we all employ to over-reduce complex decisions to something simple to easy to see based on prior or perceived outcomes. Yes, perhaps this silly example of a car arriving at work is in the stupidity bracket, but there really are 4 possible outcomes depending on the quality of a decision and the data and the outcome that you have. I mean, if you had no knowledge of what the traffic lights did and their function in your safety, you may be drawn to make this ill-informed choice. You wouldn't be aware of the other possible outcomes with their associated increased chance of accidents because you didn't have access to that data. The impact of bad decisions in cell line development and upstream is tangibly felt across projects and organizations. Beyond the obvious effects of projects progressed or cancelled incorrectly, it can also impact downstream processes. Progression of a suboptimal cell line can materially affect the cost of goods in manufacturing or force decisions to alter scale to counter issues with product quality or yield in later stages. To make predictions of future outcomes, you need to be able to have contextualized data available at the point of making the decision. Now, in science, this is well understood. But in practice, this means doing more experiments, testing more samples in a wider range of techniques. And the outcome of this is it can create a data deluge and issues making decisions on siloed or unavailable data at the time of decision making. And typical coping strategies tend to lead towards quick decisions, due simply to the difficulty of accessing data or related experimental work that you either aren't able to access or don't know it exists. Something as simple as plotting the results of 2 parallel experiments in an attempt to avoid bias can result in hours of data movement or data entry. Further, research has already shown that the effect of bias on decision making has been shown to be far more severe in the cases when it's associated with people of higher cognitive ability. The likelihood is that your organization and the projects that you're working on are being impacted by biased decisions driven by missing or unavailable data because barriers exist to the access, the presentation, the searching, the filtering needed to gain insight to make a good decision or an earned outcome. During 2020, we interviewed a range of top 20 pharma and biotech organizations in biologics development across upstream and cell line development. And we found a consistent number of users who found it extremely hard to get access to data from the multitude of systems in a timely and traceable way. Many were repurposing systems not designed to help make decisions and covering gaps by creating in-house offerings or simply using a plethora of Excel templates. This is probably why 30 to 40% of time in laboratories is being spent on documentation and administration, and 10% of rework is being based on data not being available, generating this rework. So firstly, it cannot be right that the decisions of multi-million-pound drug projects are being made on data buried in Excel templates, scattered across USB sticks and shared drives and emails. If we make the data hard to get to, we will simply drive to simple conclusions and will end with biased outcomes and trusting to somewhat to luck. Secondly, I argue that this disconnected environment is creating a poor user experience. of heavily customized offerings glued together with in-house software patches, we're making it really hard to do things easily. So yes, it's important to have accessible data. But when data is accessible, it's often in a format that's really hard to work with. Pulling CSV files of systems, aligning, correcting different nomenclatures, tidying data up. Just for a moment, and I know we're all stuck in our various locations around the world, trapped to some degree by COVID, but in a normal time when we would be trying to book a holiday, would we go about booking a holiday like this, receiving a CSV from our insurance company, an Excel spreadsheet from different hotels, an XML document from our hotel, from our flight booking facility? It would be impossible for us to actually make decisions. The expectations that we now have of easy to use and navigatable software is much higher than it was 10 or 20 years ago. Excel, after all, was created in 1987, 33 years ago. And yet it remains the case that instrument data are being manually transcribed into departmental Excel templates. And we have found, when we've reviewed that there is inconsistency from site to site, group to group, on key concepts from IDs and naming conventions to sometimes secondary calculations. And worse still, those Excel templates are being edited from person to person, from project to project. When you consider that the process of how a biologics is made and where it comes from are essential, it is really important to capture why choices were made, tracking the outcome back to the decision and back to the data that was available to make that decision. So yes, it's important to have access to data and the context with which the decision was made, but it's also really, really important that the data presentation is done in a way that's helpful. It's not just a question of getting more data unless it's accessible and usable. And I think we've all been burdened over the years with systems or tools that give you an infinite number of scrollable columns, or even worse perhaps, scatter plots of differently colored blobs, sized and shaped differently, overlaid on top of each other in an impenetrable and often difficult to navigate way, not really helping you to make a decision, but just displaying a large amount of data. This is no way to use data to make decisions, and there is a better way. In biopharmaceutical development, time is being lost to paper, and to Excel, and to ad hoc systems, and this poses operational challenges, wasting time, impeding process, jeopardizing understanding and quality. Our own research has heard that scientists and process engineers estimate they spend as much as half a day of their workday finding and reconciling and assembling data. Any delay in the development has meaning and real-world consequences. Whilst unmet medical needs remain unmet and the opportunity to recoup development costs against IP exclusivity is being lost, the cost impact of shortened time to market isn't linear, and data presented in context and consistency in execution around that data can reduce rework by up to 30%. The absence of process understanding introduces risks in low-yielding unit operations. If you consider that a typical biologic drug has a market value of greater than $10,000 a gram, these costs aggregate together really rapidly, particularly in the downstream ends of development. And this cost is being compounded by the high-risk nature of biologics development. in the context of all of these risks, these operational burdens that I talk about occur in two forms. The first is the inability to understand root cause when undesired or unexpected outcomes occur. And the second is insufficient data support, reducing the ability to make informed and timely decisions as a therapeutic candidate is being progressed. So the appeal I am making is that if you have access to real-time data, then you can gain insight. You can rank and sort process parameters by importance, for example. You can algorithmically rank clone cell lines. You can determine which best meet the performance and quality characteristics that you desire. The data is ever more complex, and it's no longer acceptable to spend time tabbing through Excel, trying to pivot data and manually run through, manually view data from systems like miniature bioreactors or metabolite sensors or product titer data. To get the best out of these high value assets, you have to have a cohesive approach to accessing, collecting, aligning, and presenting the data so you can make informed decisions. But you also need to go further in informing and enabling deep dive into data sets so that different hypotheses can be tested to ensure that more possible outcomes are being explored at the point of making key decisions. Simple user experience can save a huge amount of time. And being able to provide simple answers quickly, whilst providing access to data for rich and powerful deep dives in to explore alternative hypotheses, is what is necessary to avoid biases in decision making. As you've heard, biopharmaceutical drug makers are really eager here to apply the power of an in silico data science future state, the much mentioned Laboratory 4.0. And they aim to bring data modeling and simulation to the development processes to shorten time to market. By adopting an informatics capability that provides a scalable, modern, open, and accessible data management layer, and enterprise analytics capabilities embedded in it, you lay the foundations of an in silico-based approach to inform and advise on outcomes buried in this complex data. By democratizing data, you enable more eyes on it, and you open it up to other data and to other opinions. And this will enable organizations and teams to make better decisions. It's a framework, essentially, that scales the demands of a data-driven laboratory through a platform and a software digital play. This ability to use real-time data from which insight and decisions can be made helps organizations to build that path towards the smart factory and laboratories, the process automation, the real-time data that I mentioned to ensure quality, the efficiencies through quality by design, which needs data and decisions around that. So, IDBS will be launching a new addition to our bioprocess lifecycle management system, Polar, to support high throughput processes. And at launch, the first capabilities that we will bring to market will include automated data capture from multiple leading high throughput laboratory equipment systems, including those widely used in cell line development and upstream. Best of breed embedded analytics capabilities designed to enable simple decision-making interfaces, as well as exploratory and deep dive analytics. And all of that built on top of scalable and accessible data management layers. It's our aim, essentially, to capture the data at the point of its generation, to preserve the context through that laboratory workflow, to provide teams with real-time insights to enable more data to be brought to bear at the point of key decisions, for example, when reviewing clones and deciding which ones to progress, or which conditions to optimize for process parameters for upstream. We'll be launching this in the first half of the year in 2021, in Q1 actually in 2021, and you'll be able to register to find out more from January 2020 on our website. So to just conclude here, I have argued that making decisions requires richer data to avoid trusting to luck. The appeal I would make to scientists at the bench is to argue to stop being a data entry clerk and be a data-driven scientist. Remove the need for manual transcription of data from laboratories. Enable scientists and teams to make decisions with rich data sets by providing modern software offerings. Save time, avoid rework from missing data, incorrect assumptions, human error. Improve the time taken to get to first in human by ensuring your analytics and data management keep pace with the laboratory automation investments you are making today. And build a foundation for effective in silico modeling by capturing the outcomes, tracking those back to the decisions, but also back, very importantly, to the data, so you can validate the quality of those decisions in future state. Thank you very much for listening, and I will take some questions now.
That's great, Matt. Thank you very much. We'll go ahead and get the recording stopped.